Where's the proof? Introduction. In recent years, the integration of advanced nanomaterials, particularly graphene and its derivatives, into biomedical research has sparked both excitement and concern. Although COVID-19 vaccine manufacturers and regulatory agencies have consistently denied the presence of graphene oxide in vaccine formulations, these assertions remain largely unverified by publicly available, peer-reviewed studies. Claims of graphene detection from unofficial sources have further fueled public skepticism, highlighting a critical gap in independent scientific oversight. This video aims to illuminate the importance of conducting rigorous, third-party verification of vaccine contents, particularly under physiological conditions, to ensure transparency, accountability, and public trust in medical interventions. Overview. While several unofficial or independent sources have claimed to detect the presence of graphene or graphene oxide in COVID-19 vaccines, no comprehensive, peer-reviewed studies have been made publicly available that directly test for and verify the absence of graphene in these vaccines. Additionally, there is no known study that examines the contents of these vaccines at body temperature, approximately 37 degrees Celsius, a critical oversight. Considering the potential for certain compounds or nanostructures to change behavior or visibility at physiological temperatures. While the absence of peer-reviewed studies verifying the presence of graphene oxide does not constitute proof of its inclusion, the continued lack of publicly available, independent studies that directly confirm its absence, particularly in light of persistent claims, raises questions. If the goal were to definitively dispel these concerns, one might expect that such a study would have been conducted and made public by now. Context and claims of graphene presence. Reports alleging the presence of graphene oxide in COVID-19 vaccines have circulated since 2021. These claims are primarily based on analyses conducted by individuals or groups outside traditional scientific or regulatory frameworks. While these studies often lack transparent methodologies, verified sample provenance, or peer review, they have nonetheless raised public concern and skepticism, particularly among those wary of new vaccine technologies or governmental oversight. Absence of studies proving graphene is not present, despite repeated public statements from vaccine manufacturers and government agencies that graphene oxide is not included in any authorized COVID-19 vaccines. No scientific studies have been made publicly available that specifically test vaccine samples for the absence of graphene compounds. Instead, the public is expected to rely on manufacturer-disclosed ingredient lists and regulatory oversight mechanisms. While this may be standard practice in pharmaceutical regulation, it does not meet the evidentiary standard expected by individuals seeking independent, empirical validation. Potential applications and dangers of graphene in vaccines. Graphene and its derivatives, particularly graphene oxide, GO, are highly researched nanomaterials with remarkable properties: extremely thin, lightweight, strong, electrically conductive, and bioreactive. These features make them attractive candidates for a wide range of biomedical applications, including drug delivery systems and advanced diagnostics. However, their use, especially in injectable products like vaccines, is still largely experimental and brings with it a number of serious concerns. Potential applications of graphene in vaccines and medicine. Targeted drug delivery. Graphene oxide has been explored as a nanocarrier capable of transporting drugs, proteins, or RNA directly into specific cells, including immune cells. In theory, this could increase vaccine efficacy by improving cellular uptake and reducing off-target effects. Adjuvant properties. Some studies suggest that graphene oxide might act as an adjuvant, a compound that stimulates the immune system and enhances the body's response to an antigen. This could lead to stronger immune responses with lower antigen doses. Diagnostic integration. Graphene's electrical conductivity could theoretically enable smart vaccines, injections with built-in diagnostic or tracking capacities, e.g., for measuring immune responses or interacting with wearable devices. Photothermal and stimuli-responsive functions. Graphene can absorb light and convert it into heat, photothermal effect, opening doors for external control of vaccine activity, though this remains speculative. Potential dangers of graphene in vaccines. Despite its promise, the use of graphene in living systems is far from fully understood, especially when injected directly into the bloodstream or muscle tissue. Toxicity and inflammation. Studies have shown that graphene oxide can induce oxidative stress, inflammation, and cellular damage in various biological systems. Toxicity may vary based on dose, particle size, surface modifications, and exposure duration, making it difficult to generalize safety. Immune system overactivation or suppression. While some hope graphene could boost immune responses, others fear it could trigger unintended immune activation, leading to autoimmune issues, cytokine storms, or hypersensitivity. Conversely, it could also suppress certain immune functions, potentially reducing the body's ability to fight other infections. Accumulation in organs. Animal studies have shown that graphene-based materials can accumulate in the lungs, liver, spleen, and brain, raising concerns about long-term exposure and delayed toxicity. Blood-brain barrier penetration. Due to its nanosize, graphene oxide may cross the blood-brain barrier, which is particularly concerning given its unknown neurological impacts. Interference with cellular processes. Graphene's sharp edges and ability to interact with DNA and cell membranes raise concerns about genetic damage or disruption of cellular communication. Lack of regulatory approval. As of now, there is no known regulatory approval of graphene or graphene oxide as an ingredient in any licensed human vaccine, and no such material appears in the publicly disclosed ingredient lists of currently authorized COVID-19 vaccines. Graphene is a cutting-edge nanomaterial with enormous potential, but also significant risks, particularly when used in intracellular or injectable applications. Its use in vaccines must be approached with extreme caution, full transparency, and extensive safety testing. Without clear consent, peer-reviewed research, and regulatory approval, the inclusion of graphene in any vaccine formulation would be ethically and medically irresponsible. What about quantum dots? Graphene quantum dots, GQDs, are nanoscale fragments of graphene that exhibit unique electrical, optical, and chemical properties, making them subjects of extensive research across various scientific domains. Their exceptional characteristics have led to explorations in fields such as energy storage, biosensing, bioimaging, and photocatalysis. Graphene quantum dots in biosensing and bioimaging. GQDs have shown significant promise in biosensing and bioimaging applications due to their fluorescence properties and biocompatibility. They have been utilized for imaging tumors and monitoring therapeutic responses, as well as detecting specific biomolecules like microRNAs and proteins. These applications leverage the photoluminescence of GQDs to provide sensitive and specific detection mechanisms. Graphene quantum dots in energy storage. In the realm of energy storage, GQDs have been employed to enhance the performance of batteries and supercapacitors. Their high surface area and electrical conductivity contribute to improved charge storage and transfer capabilities, leading to devices with higher efficiency and capacity. Graphene quantum dots in photocatalysis. GQDs have also been integrated into photocatalytic systems, such as TO2 GQD nanocomposites, to improve light absorption and charge separation. This integration enhances the efficiency of photocatalytic reactions, which is beneficial for applications in environmental remediation and energy conversion. Below is a speculative analysis based on the known properties of graphene quantum dots, GQDs, and how they could be applied, especially in emerging fields like biointerfaces, data transmission, biosurveillance, and decentralized computing systems such as blockchain or cryptocurrency mining using biodata. Speculative analysis. Potential uses of graphene quantum dots, GQDs. GQDs are nanoscale particles derived from graphene sheets, known for their exceptional conductivity, quantum confinement, photoluminescence, chemical reactivity, and biocompatibility. These traits make them powerful candidates for a wide range of advanced technological applications, including theoretical uses that have yet to be fully explored or confirmed by mainstream science. Animal studies. Several animal studies have explored the potential of GQDs in biomedical applications. In vivo imaging, research has demonstrated the use of GQDs for in vivo fluorescence imaging in animal models, highlighting their potential for real-time tracking and diagnostic purposes. Toxicity assessments. Multiple in vivo studies have been conducted to evaluate the toxicity of GQDs. For instance, a study involving mice investigated the effects of multiple GQD injections, providing insights into their biocompatibility and safety profiles. Although GQDs have shown promise in preclinical studies, particularly in animal models, their application as implantable or injectable biosensors in humans remains largely in the experimental stage. Ongoing research continues to address challenges related to their safety, efficacy, and regulatory approval to facilitate their transition into clinical settings. Biometric interfaces and biosurveillance. Speculative use. GQDs could be used as implantable or injectable biosensors, capable of interfacing directly with human tissue to detect, process, and transmit biological data in real time. Why it's possible. GQDs fluoresce in response to environmental changes like pH, temperature, or the presence of biomolecules. They can be functionalized to bind to specific cellular components. Their size allows them to move easily through biological systems. Implications. Continuous real-time monitoring of health markers like glucose, cortisol, neurotransmitters, or even emotional states. Potential integration with biosurveillance systems via wireless communication protocols, e.g., 5G or near-field communication. Data harvested from individuals could be stored in health databases or used for predictive diagnostics. Human studies. Direct studies involving injectable or implantable GQD-based biosensors in humans are currently limited. However, related advancements in graphene-based technologies indicate progress toward clinical applications. Graphene-based brain implants. A notable development is the clinical trial of a graphene-based brain-computer interface, BCI, device in the UK. This implant aims to differentiate between cancerous and healthy brain cells by detecting their electrical emissions, potentially enhancing the precision of brain tumor surgeries. While this BCI device does not specifically utilize GQDs, it exemplifies the translational potential of graphene-based materials in human medical applications. 5G enabled nanonetworks. Speculative use. GQDs could act as nodes in a wireless nanosensor network, potentially powered or activated by 5G electromagnetic fields. Why it's possible. GQDs are conductive and responsive to electromagnetic frequencies. Their quantum properties make them suitable for ultrasensitive signal processing. In theory, they could convert biological signals into transmittable data over short distances. Implications. Wireless health monitoring systems embedded in the body. Potential interface with smart environments, e.g., smart homes, wearable tech, autonomous medical response systems. Raises concerns about remote access to internal biological data without consent. A study found in the International Journal of Nanoelectronics and Materials by Saidal Hamida Asterisk and Go-Chin Hawk entitled Development of 3.5 GHz Enhanced Graphene Patch Antenna for 5G Applications directly supports the feasibility of using graphene-based materials and, by extension, GQDs, in systems operating at 5G frequencies. While not a biological or biomedical study, it proves that graphene composites can resonate efficiently at 3.5 GHz, which is crucial for the development of injectable or implantable biosensors that might leverage 5G for real-time data transfer or external activation. Biometric crypto mining and data monetization. Speculative use. GQDs could be used to harvest bioenergetic or biodata signals from the human body, then feed those into blockchain or cryptocurrency networks as a form of microtransaction mining. Why it's possible. The body generates measurable electrical activity, EEG, ECG, etc., which could be captured and quantified. GQDs could potentially transduce these signals into digital patterns or keys. With secure processing, biometric signatures could be used for transaction verification or data monetization. Implications. Individuals may become miners by simply existing, producing valuable biodata for corporations, insurers, or governments. Raises major ethical questions. Who owns the data? Who profits? Is consent informed and voluntary? Could lead to human as a platform economic models, blending health tracking, predictive AI, and financial systems. While these applications may seem futuristic, the trajectory of corporate research suggests they are already being explored. For example, Microsoft has been granted International Patent W20200606066A1, which outlines a system where human body activity data is used to mine cryptocurrency, effectively making biodata a commodity. Although not yet implemented in medical devices or vaccines that we know of, such patents reflect a clear interest in merging biology with digital finance and surveillance infrastructure. Programmable nanomedicine and remote activation. Speculative use. GQDs could be used in programmable drug delivery systems where specific frequencies, e.g., from a 5G tower or handheld device, activate or modulate the release of medication within the body. Why it's possible. GQDs are sensitive to light, heat, and electromagnetic fields. Functionalized GQDs could be attached to drug molecules and only release them upon receiving a specific signal. Could allow for external control of internal biological responses. Implications. Tailored, on-demand treatment protocols without repeated injections. Potential for misuse or unauthorized activation. Surveillance or manipulation of physiological states becomes conceivable, raising significant bioethical concerns. Quantum encryption and identity tagging. Speculative use. Each person could be tagged with a unique quantum signal, or ID, using GQDs that fluoresce in a distinct spectral signature, potentially enabling unforgeable biometric verification. Why it's possible. Quantum dots can be manufactured with highly specific emission spectra. These biotags could, in theory, provide individual-specific identifiers. Implications. Could be used for security, vaccine verification, border control, or digital identity. Also raises the possibility of permanent tracking or identification without consent. May interface with digital ID systems or centralized databases. Conclusion. While the speculative uses of graphene quantum dots lie at the edge of what is currently possible, their foundational properties absolutely support these futuristic applications. As science continues to push the boundaries between biology, data, and machine learning, GQDs may become central to the development of nanobiodigital interfaces. However, the line between innovation and intrusion is thin. Without strict ethical frameworks, privacy protections, and clear informed consent, these technologies could easily be abused for surveillance, behavioral manipulation, or exploitative economic models. The rapid deployment of medical technologies under emergency authorization, such as during a global pandemic, can create a unique window wherein experimental innovations are implemented without the usual degree of scrutiny or long-term safety data. In such scenarios, novel materials like graphene quantum dots could theoretically be introduced under the guise of urgency, bypassing the full regulatory rigor typically required for new biomedical applications. While intended to save lives, this accelerated pathway also opens the door for technologies still in early experimental phases to be deployed at scale, potentially without the public's full understanding of their function, implications, or long-term effects. While regulatory agencies require full disclosure of ingredients, they rely on manufacturers to provide both the data and the samples for analysis. Independent lab testing, where it exists, is typically performed on manufacturer-submitted vials, not on doses pulled from real-world clinical settings. As such, there is no public assurance that the sample analyzed by regulators is compositionally identical to what is stored in a doctor's refrigerator or administered in a vaccine clinic. If a manufacturer knew that a particular ingredient, such as graphene, was not approved for use, it would be logical for them to exclude it from any regulatory sample, even if it were present in distributed batches. This scenario, while hypothetical, illustrates why independent, Random sample testing of distributed vials is essential to ensure product consistency, safety, and public trust. The unacceptability of contaminants in vaccines. The presence of any unapproved or undisclosed substances, often referred to as contaminants, in vaccine formulations is categorically unacceptable, regardless of whether they are believed to be harmful or inert. Concerns raised about the possible presence of graphene in vaccine samples have often been dismissed by authorities as either misidentifications or unknown contaminants. Terminology that effectively acknowledges the detection of unclassified materials without offering meaningful investigation or transparency. This pattern of dismissal sidesteps the core issue that any undisclosed, unidentified substance in a biologically active product is unacceptable, regardless of whether it was intentionally added or inadvertently introduced. Vaccines are biologically active products administered to healthy individuals, including vulnerable populations such as children, pregnant and nursing mothers, the elderly, and those with compromised immune systems. As such, they are held to some of the highest purity and safety standards in medicine. Allowing or excusing the presence of contaminants undermines the fundamental principles of pharmaceutical manufacturing, including regulatory compliance. All ingredients in vaccines must be reviewed and authorized by regulatory bodies such as the FDA in the US, the EMA in the EU, or MHRA in the UK. The presence of any unlisted substance, even in trace amounts, would constitute a breach of these legal and regulatory frameworks. Informed consent. Patients have the right to know what is being introduced into their bodies. The discovery of any undisclosed material invalidates the ethical standard of informed consent and could expose manufacturers or medical providers to legal liability. Quality assurance and batch consistency. The presence of contaminants could indicate lapses in the manufacturing process, sterilization protocols, or supply chain integrity. This calls into question the consistency and safety of other batches, even if only a few are reportedly affected. Risk of unknown biological interactions. Even if a contaminant is not immediately harmful, its interaction with biological tissues, immune responses, or other medications is unknown. Long-term effects cannot be predicted without specific safety testing, which is not conducted for undeclared materials. Public trust and ethical responsibility. Allowing contaminants to go uninvestigated or unacknowledged erodes public trust in health authorities, manufacturers, and the scientific process itself. Public health efforts rely not only on efficacy and safety, but on transparency and accountability. In summary, no level of contamination is acceptable in injectable products designed for mass immunization. Any credible study that identifies foreign substances, such as graphene oxide, metals, or unidentified particulates, demands a full, transparent investigation by regulatory authorities, followed by public disclosure of findings and corrective measures. Lack of analysis at body temperature. Equally concerning is the apparent absence of any published studies that analyze the behavior or composition of the vaccine at body temperature. Since some nanomaterials, including graphene derivatives, are known to respond to thermal changes, it is scientifically important to understand whether any structural transformations or emergent properties occur under physiological conditions. This is particularly relevant for materials suspected of operating at the nanobiointerface. Why these issues matter. Scientific integrity and transparency. The scientific method requires not only proof of presence, but also valid procedures to confirm absence. Without transparent, independent studies demonstrating that graphene oxide is not present, claims of its exclusion remain unverifiable to the public. Public trust. For trust in public health interventions to remain strong, transparency and independent verification are essential. Relying solely on manufacturer-provided data without third-party testing accessible to the public has fueled doubt and suspicion. Behavior under physiological conditions. Body temperature can influence the behavior of materials at the molecular and nanostructural level. If graphene oxide or any similar nanomaterial were present, understanding its behavior at 37 degrees Celsius would be critical to assessing potential biological interactions. Informed consent. A core tenet of medical ethics is that individuals should be able to give informed consent. Without a full accounting of vaccine contents verified by independent analysis, the consent given may be considered insufficiently informed by some. To date, no peer-reviewed, publicly accessible scientific studies have been published that conclusively demonstrate the absence of graphene or graphene oxide in COVID-19 vaccines, nor have any such studies been conducted under body temperature conditions. Given the potential implications and the level of public concern, these gaps highlight a need for transparent, third-party verification that goes beyond regulatory assertions and aligns with the principles of scientific rigor and public accountability. The risk of solely relying on regulatory agency statements. While regulatory agencies such as the FDA, EMA, or MHRA play a critical role in ensuring the safety and efficacy of vaccines, relying exclusively on their statements without independent verification introduces significant risks, especially in a system where the chain of custody and source of tested samples is controlled by the manufacturer. Selective sample submission vaccine manufacturers are typically responsible for providing samples for regulatory review and quality assurance. If oversight is not thorough or randomized, there exists the possibility, whether accidental or intentional, of submitting only pristine, specially prepared batches that do not reflect the full scope of what is being distributed to the public. This creates a loophole in accountability and calls into question the integrity of batch-level consistency. Conflict of interest and regulatory capture. In some cases, regulatory bodies can become subject to regulatory capture, where the interests of the industry they are meant to regulate begin to unduly influence their decisions. Funding structures, revolving doors between regulators and industry, and political pressures can all contribute to a situation in which the appearance of oversight is maintained without sufficient depth of scrutiny. Lack of independent cross-verification. Scientific integrity depends on reproducibility and transparency. Without independent laboratories confirming the findings of regulatory agencies using randomly selected, off-the-shelf vaccine samples, there is no way for the public or the broader scientific community to validate official claims. Transparency is the cornerstone of public trust. Secrecy or over-reliance on centralized information erodes it. Historical precedent. History has demonstrated that regulatory agencies, while essential, are not infallible. Incidents involving unsafe medications, e.g., thalidomide, Vioxx, were permitted under regulatory regimes that initially failed to detect or disclose critical safety information. In each case, independent research and whistleblowers played pivotal roles in revealing the truth, highlighting the need for distributed accountability. Ethical implications. The ethical standard for public health interventions, particularly those administered en masse, demands that no single entity should be the sole gatekeeper of safety data. A system that is fully transparent, open to third-party review, and subject to meaningful checks and balances is essential to avoid unintended harm and to maintain informed public consent. In short, while regulatory agencies are necessary, their conclusions should never be treated as beyond question. True safety and public trust require independent oversight, random sampling, full transparency, and rigorous scientific inquiry that is open to scrutiny from professionals outside the regulatory structure. Financial relationships between regulatory agencies and pharmaceutical companies. A significant concern in the modern regulatory landscape is the financial entanglement between pharmaceutical companies and the very agencies tasked with regulating them. In many countries, including the United States and parts of the European Union, a substantial portion of regulatory agency funding comes from the pharmaceutical industry itself, raising unavoidable questions about independence, objectivity, and potential conflicts of interest. User fees and industry funding. Agencies like the U.S. Food and Drug Administration, FDA, receive billions of dollars annually in user fees from pharmaceutical companies under legislation such as the Prescription Drug User Fee Act, PDUFA. These fees are intended to expedite the drug approval process by supplementing the agency's budget, but they also mean that pharmaceutical companies are effectively paying for their products to be evaluated. In 2022, User fees accounted for more than 45% of the FDA's drug regulatory budget. In the case of the European Medicines Agency, EMA, industry fees cover roughly 90% of its operating budget. Perception of bias and eroded trust. Even if regulatory decisions are made with integrity, the public perception of bias becomes difficult to overcome when the regulated parties are also the primary source of funding. The inherent conflict of interest can lead the public to suspect that safety assessments may be influenced by commercial interests, especially when there is a strong financial incentive to approve or maintain approval for a widely used product. Faster approvals, less scrutiny. User fee agreements often come with performance goals, such as shortening review times for drug and vaccine applications. While faster reviews may seem efficient, they can also lead to rushed assessments and reduced scrutiny, especially when agencies are under pressure to meet industry-set timelines. Accountability and oversight gaps. When regulatory agencies depend on industry funding, there's a risk that they may become less willing to challenge powerful sponsors or rigorously investigate claims of contamination, adverse effects, or manufacturing inconsistencies. This risk is compounded when independent verification and external auditing are absent. This dynamic doesn't mean all approvals are invalid, but it does mean we should be wary of treating agency assurances as the final word without third-party, independently funded research to confirm their claims. Recommended protocol for independent verification of vaccine contents. In order to establish definitive public trust in the safety and purity of COVID-19 vaccines or any injectable product, a transparent, replicable, and decentralized testing protocol must be implemented. The following steps outline what must happen moving forward. Testing of distributed vaccine vials, not manufacturer samples. Analysis must be conducted on vials drawn from real-world distribution, meaning those sent to pharmacies, clinics, hospitals, or individuals for human consumption, not samples prepared and submitted by the manufacturer. This ensures that what is tested reflects what is actually being injected into the public. Analysis at body temperature. All testing should include analysis at physiological temperature, approximately 37 degrees Celsius, 98.6 degrees Fahrenheit. Some compounds, particularly nanomaterials, may behave differently or become detectable only at body temperature. Testing at room temperature alone is insufficient to rule out the presence or transformation of certain substances under biological conditions. Full spectrum analysis of contents. Each sample must undergo a full spectrum materials analysis, including microscopy, optical, electron, and scanning probe. Spectroscopy, Raman, mass IR, UV-Vis. Chemical assays. Nanoparticle analysis. Biological activity observation at body temperature. Documentation and identification of any contaminants. If any unlisted or unexpected particles, materials, or substances are detected, they must be photographed, recorded, and measured using verifiable instruments. Their chemical composition and structure must be identified to the greatest degree possible. Investigators must attempt to trace the origin of the contaminant, whether from laboratory, manufacturing, packaging, storage, transportation, or deliberate addition. Split sample verification by an independent lab. Following initial analysis, the remainder of the vial must be immediately sealed and sent to a separate, unrelated laboratory for verification. This ensures results are reproducible. Data is not influenced by individual researcher bias. No tampering has occurred. A chain of custody can be properly documented. Full public disclosure of results. All findings, including methodology, lab credentials, raw data, images, and analytical results, must be made freely available to the public for review and scrutiny. If contaminants are found, appropriate regulatory bodies must be notified, and a public inquiry into the source and scope of the contamination must be initiated. This process does not require political permission or corporate cooperation, only scientific integrity, transparency, and courage. It is the next logical step for any honest society seeking truth, safety, and public trust in its medical systems. Legal and ethical accountability beyond pharmaceutical companies. While many pharmaceutical manufacturers have secured legal indemnity, often granted under emergency use authorizations or through government agreements, this does not extend immunity to all parties involved in the distribution and administration of these products. Healthcare providers, public health agencies, and even private distributors do not automatically share in this legal protection. If future evidence reveals that individuals were harmed due to undisclosed ingredients or contamination, particularly if those materials were present in vials administered under the assumption of full disclosure, those who facilitated or enforced distribution may be held liable, both civilly and, in some cases, criminally. Furthermore, legal indemnity does not equal moral immunity. The duty of care, ethical responsibility, and commitment to informed consent remain intact for all involved in public health. Knowing participation in the distribution or administration of a product that contains undeclared, untested, or unauthorized substances could potentially result in personal or institutional liability, especially if warnings were ignored or testing was actively discouraged. In short, the absence of manufacturer accountability does not eliminate the responsibility of others. It increases the need for vigilance, transparency, and independent verification from every other actor in the system. Conclusion. In the absence of publicly available studies that confirm the non-existence of graphene oxide in COVID-19 vaccines, especially under conditions that reflect how the vaccines function in the human body, public trust cannot be fully restored through official assurances alone. The potential applications and risks of graphene-based nanotechnology, combined with historical and ongoing concerns about regulatory conflicts of interest, call for a new era of scientific transparency. Independent, decentralized verification of vaccine composition is not only prudent, it is necessary. Without it, informed consent, ethical responsibility, and legal accountability remain compromised. It is the responsibility of an honest, scientifically grounded society to demand and conduct such verification in the service of truth, safety, and public health.